Today lesson by Python lesson 1. We need the BioPython install the computer. Then I show you this code for install BioPython. We already installed my computer the BioPython. Import bio, print bio version. We check up this code. In this code, Bio.Seq and Seq are part of the BioPython library. 1. BioSeq, Bio.Seq. Used to call the Seq module of the BioPython library, this module can be used to sequence nucleotide or amino acid sequences. Allows you to process sequences. 2. Seq. The Seq class is BioPython. This class represents DNA, RNA, or protein sequences and performs various operations on them. Allows you to perform operations, for example, finding a complementary sequence or reversing. Sort my Seq and so my Seq equals Seq. Here a variable called my Seq is created and the nucleotide sequence AGTACACTGGT is assigned to it. The sequence is converted to a Seq object, which means it is now a special object that can be manipulated by BioPython's functions. 4. Print my Seq. This code prints the contents of the mySeq variable to the screen, meaning you get the result AGTACACTGGT. This is Seq converts DNA, RNA, protein sequences that appear as plain text into a special object for processing with BioPython functions. This object can perform various operations such as finding the complementary strand of DNA, transcribing and reversing. It is possible to carry out. This is for complementary of sequence RNA. RNA no. DNA for DNA sequence. This for DNA or RNA or protein for use. Okay. Complement. In the code, the mySeq.complement function is a method of the Seq object of the BioPython library and returns the complement of a DNA sequence. Returns the complementary chain. How does it work? It is work. In DNA, each nucleotide pairs with the complementary base opposite it. We know adenine, thymine, guanine, cytosine. The working principle of the code is as follows. If my Seq was initially defined like this: from Bio.Seq import Seq my Seq agtacact print my Seq that complement. Here, when the mySeq complement function is called, each nucleotide is replaced with its corresponding complement. Result: the complementary sequence printed on the screen. Sequence TCATGTGACCA. Okay, this. As a result, the mySeq complement method returns the complementary strand of DNA. This method is used in bioinformatics, especially in genetics. It is often used in assays and PCR design. Only for DNA sequence. Just DNA sequence. This not this, yes. Only DNA sequence. But mySeq reverse complement use maybe DNA, RNA, RNA, or protein. Okay. So, what does the mySeq that reverse complement method do? mySeq reverse complement is a method of the Seq object in BioPython. This method returns the complement of a DNA sequence, returns the complementary and reverse chain. What is the difference? Complement returns the complementary strand of a DNA sequence. Reverse complement first finds the complementary strand, then reverses it. Code execution processes. Okay. This complement, this reverse complement. Okay. This prints. Okay, then. This this Python code reads a FASTA formatted bioinformatics sequencing file using the SeqIO module of the BioPython library and outputs the following information. 1. The ID number, for example, ID number of the sequence, DNA or protein sequence. Second sequence length, for example, len function, len len method. Sequence record. We have record of DNA sequence, RNA sequence, or protein sequence. Amino acids. The length of the sequence And sequence repr sec record sec. Okay, print the nucleotide sequence. Write the file name correctly um, for work. Okay, so um, SeqIO. This is module used to read and write sequencing files from the BioPython library. File path. File path indicates the full path of that FASTA format file containing sequence data. And for sec record in SeqIO for sec record in SeqIO dot parse file path FASTA. Okay. The file path indicates the full path of that FASTA format file containing sequence data. R, 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 the yes, these are raw string named called used to prevent characters from being treated as special characters. And for sec record in SeqIO that parse file path FASTA. Okay, so I explain to you. SeqIO that parse reads the given file in FASTA format. For sec record in, it processes each sequence record, for example, maybe call it, in the file one by one. Okay. Oh, also, this we know what is to the work. Okay. Code execution result ID sequence length and sequence and this sequence. Also we use yes work, but sequence executed, but we don't want the executed of sequence. We need short sequence of wife. Okay. Okay. So then, mm, bioinformatics use of code, read genomic or pattern sequences in FASTA format, obtaining sequencing data for genetic analysis, checking the lengths of DNA or protein sequences, automation of sequence processing and analysis processes. Note: FASTA format is widely used in bioinformatics to store DNA, RNA, or protein sequences. SeqIO.parse function is used for reading only if the sequence is to be modified. It must be rewritten with SeqIO that write function. Okay, this code this code uses for GenBank format sequence. Okay. Explanation: this Python code reads a GenBank format, maybe call it .g, .gbk sequence file using the SeqIO module of the BioPython library and outputs the following information: 1. Sequence ID number, sequence length, sequence itself. Okay, then bioinformatics use of code read genomic or protein sequences in GenBank format, obtaining sequencing data for genetic analysis, automate sequence processing and annotation processes, get additional metadata, gene name, features, annotations from a GenBank file. Feature: data type format, supported data, and two GenBank and FASTA. Compare. We compare data format. GenBank uh, give us genetic sequencing plus annotation. FASTA only sequencing. GenBank give us multi-line structured. FASTA and simple and compact. GenBank give us sequence gene annotation property source. FASTA give us sequence and title. For example, ID. GenBank of abbreviation. GenBank. FASTA fast. Okay, then this code explained by me. From Bio.Seq, import Seq, this code building a loop on nucleotides. For index letter enumerate, for example, mySeq, print index letter. Enumerate enumerate this. Retrieves each nucleotide of the sequence along with its index. For index letter in, cycle returns an index and a letter for each nucleotide. Print index letter for this. Mhm. Print index letter. Um, Prints each nucleotide and its index to the screen. The first 5 lines print each nucleotide in the sequence and its index number. The last line displays the total length of the sequence. Bioinformatics use of code processing individual nucleotides and amino acids in DNA, RNA, protein sequences, analyze the length and composition of the sequence, perform iteration operations in bioinformatics algorithms use the same methods as by Python strings when working with sequences. This code is a simple example for analyzing DNA sequences and can be used for more advanced, can be the basis for bioinformatic operations. Okay, and my seg this code stride um, len my seg returns the number of nucleotides in the sequence, and len this len method. Okay, this returns we returns us 20, and because the sequence consists of 20 nucleotides. Okay, um, G contains G counts how many times the nucleotide G is repeated in the sequence. For example, if there are 20 G's in the sequence, the result will be 20. My seg this for divide 12, print my seg for divide 12. This code for print my seg, this code selects the section starting from index 2 4 to index 12, not including 12. Since Python indexing starts at 0, the fourth index will be the letter A, because 0, 1, 2, 3, 4, this A returns us. Okay, selecting part of a sequence with certain steps, stride this. Okay, my seg 3 and 3 divide 2 starts from index 3, letter A, 0, 0, 1, 2, okay, and 3. Letter A creates a new sequence by skipping 2 indexes each time. For example, basic sequencing G C G T A A C A A G G T T T T C C G T A G G T G A A C C T G C blah blah blah. This code, this sequence returns A A A A G G T no, sorry, AAG returns uh, by 3 divided by 2 this code, and TTC, and reverse the sequence, completely reverses the sequence. Okay, for example, reverse prints GC content as a percentage, correct function for GC content calculation. Um, and I explain code usage areas, distinguishing certain sequences in genetic analysis, investigating different parts of DNA sequences, convert sequences for reverse complementary analysis, determining the number of individual nucleotides in genome studies. This code calculates the GC percentage of a DNA sequence using the Seq and SeqUtils models of the BioPython library. What is GC content? It represents the total percentage of guanine and cytosine nucleotides in a DNA sequence. GC content provides information about the stability and evolution of DNA. Code usage areas, calculation of GC content in genetic analysis, learn if a DNA is stable, higher GC percentage, more stable DNA, analysis of the evolution and adaptation of microorganisms, primer design in PCR, and other genetic engineering applications. Concatenating sequence. This code generating a list of DNA sequence. List elements ACGT, AACC, TTCC. Note sec ACGT. This is not a str type, but a by Python sec object. Creating an empty sequence, for example, concatenate equal sec. Concatenate an empty sec object for the sequences to be concatenated. If we want to print the concatenated sequence, we need to add these to the end of the code. Print concatenated sequence, concatenated application areas of the code, combining multiple DNA sequences, creating primers for genetic analysis, gen splicing, and mutation studies. And then, and this correct, we must correct. Okay. And okay. So then this is wrong. This is the wrong approach. This code the following steps occur in the code. 3 different Seq objects are created in a list called contigs. SeqATG, this is a start codon sequence. SeqTTCC, this is another short-term sequence. SeqTTGCA, this is another sequence. A Seq object is created in a variable called spacer, which consists of 10 n sequences. For example, this code. And where the letter n represents the unknown basis, represents with the command spacer.join contigs, the spacer sequence is joined to all sequences in the contigs list, this join operation, in concatenate sequences and adds a break of 10 n between each sequence. As a result, a new sequence is created that includes 10 n letters between the contigs. This method is used to store genetic information or useful for integrating other data used in bioinformatics. Then this code uses the Bio.Seq module of the BioPython library to show how a DNA sequence is represented in different cases. Below is an explanation of each command. DNA-seq equals seq ACGT or ACGT, capital or non-capital letter. This line creates a Seq object and stores the sequence ACGT or ACGT in a variable called DNA-seq. Here inside the Seq object there are lowercase or uppercase. There is a DNA sequence shown in capital letters. Yes. Print DNA-seq. This command prints the DNA-seq variable to the screen. The result will be the sequence ACGT as is. Print DNA-seq upper. This command converts all letters in the DNA-seq sequence to uppercase. The upper method converts lowercase letters in my sequence, replaces with uppercase letters. As a result, ACGT will be printed for this. uppercase, all letters, sequence, lowercase, all lowercase of sequence, and um, 50-50. Start lowercase and uppercase letters. Okay, then conclusion of all about this course by Python of first course. This by Python library of module and Seq also module and my Seq we are my sequence variable this sequence and print my sequence complement and print my Seq reverse complement. Final, the end. Thank you for attention. See you next lessons.